Hello friends, today in this video I am going to discuss about the MCQs on general microbiology, and this is the 17th part of the series of 500 MCQs on microbiology. So let's start the question answer session on microbiology with detailed explanations. So the first question is bacterial cytoplasmic membrane is a unit membrane composed of phospholipids and proteins freely permeable to all nutrient compounds, a site of respiratory electron transport system known to confer osmotic protection and gram staining characteristics. And the right answer is option B. That means 1 and 3. 1 and 3 is the right answer. Okay, so move on to the next question, that is bacterial cytoplasm is a colloidal system of viscous watery solution containing chloroplast, ribosomes, mitochondria, organic and inorganic solutes. And the right answer is option C. That means 2 and 4. Option 2 and 4 is the correct answer. So next one is organisms with defective cell wall include protoplast, L forms, spheroplast, mycoplasma. And the right answer is option A. That is 1, 2 and 1, 2 and 3 is the right answer. And here we will see the bacterial L form. L form means they lack the cell wall. Okay, so two type of L forms are present, that is protoplast and spheroplast. So if the gram positive organisms lack the cell wall, then it will be called protoplast, and if the gram negative organism lack the cell wall, then it will be called as spheroplast. Okay, so move on to the next question, that is following statements are concerning the surface structure of bacteria. So peptidoglycan is thinner in gram negative than in gram positive bacteria. Lipopolysaccharide are found in both gram positive and gram negative cells. Pili mediate interaction of bacteria with mucosal epithelium. Bacterial flagella are non-antigenic as they have the same chemical composition. And the right answer is option B. That is option 1 and 3. 1 and 3 is the correct answer. So move on to the next, that is L forms of bacteria are known to develop spontaneously or in the presence of penicillin. Cells that have lost the ability to synthesize peptidoglycan, spherical regardless of their original shape, or cell which require hypertonic involvement. And the right answer is option E. That is all the options are correct. Okay, so move on to the next, that is pleomorphism is commonly seen in Staphylococcus aureus, Corynebacterium diphtheria, Escherichia coli, or Haemophilus influenzae. Right answer is option C. It is 2 and 4. Corynebacterium diphtheria and Haemophilus influenzae is the correct answer. So next one is cell wall of bacteria can be demonstrated by plasmolysis, electron microscopy, micro dissection, or gram staining. And the right answer is option A. That is plasmolysis, electron microscope, and micro dissection is the right answer. All three are right answer. Next one is fimbriae are appendages of bacteria. They are organs of addition found in gram negative organisms, detectable by hemagglutination method or non-antigenic. So the right answer is option A. That means first three options, that are the organ of addition found in gram negative organism, detectable by hemagglutination method. So these are the characteristics of fimbriae. Okay, so move on to the next, that is bacterial flagella and pili originate from cytoplasm, originate from cell wall, are extensions of bacterial capsule, originate from cell membrane. Right answer is option D. That is only 4. That is bacterial flagella and pili originate from cell membrane. Okay, next one is mesosomes are a kind of ribosome associated with cell division, a part of cell wall, principal site of respiratory enzyme. Right answer is option C. That is 2 and 4. As these mesosomes are associated with cell division and principal site of respiratory enzymes. So this is the mesosome, so which is attached to the plasma membrane of the bacteria. Okay, so move on to the next, that is functions of bacterial capsule include uh, serves as a protective cover associated with bacterial adherence, enhances bacterial virulence, act as storage of energy. So the right answer is option B. That is 1 and 3. So this bacterial capsule serve as a protective cover and enhance bacterial virulence. Okay, Move on to the next question, that is which are capsulated bacteria? Bacillus anthracis, Yersinia pestis, Haemophilus influenzae, Pseudomonas aeruginosa, and the right answer is option A. These first three options are right. So capsulated bacteria, Bacillus anthracis, Yersinia pestis, and Haemophilus influenzae. Okay, so move on to the next one, that is true statement regarding bacterial capsules include protect bacteria by inhibiting phagocytosis. Capsules can be purified and used as vaccine. Capsules are produced by gram-positive as well as gram-negative bacteria. Capsules are useful in serotyping. So the right answer is option E. That is 1, 2, 3, 4. All options are correct regarding bacterial capsule. So next one is formation of very large prominent polysaccharide capsule is a feature of Neisseria meningitis, Klebsiella pneumoniae, Salmonella typhosa, or Streptococcus pneumoniae. And the right answer is option C. It is 2 and 4. That is Klebsiella pneumoniae and Streptococcus pneumoniae is the right answer. Next one is bacterial capsule can be demonstrated by negative staining, Albert staining, Coulomb reaction, or microcytotoxicity test. And the right answer is option B. That is 1 and 3. Albert stain and microcytotoxicity test is used for bacterial capsule staining. Okay, so next one is extracellular visible polymeric substance surrounding the cell wall of bacteria are capsules, slimes, microcapsule, or outer membranes. And the right answer is option A. That is first three option, that is capsules, slimes, and microcapsules are the right answer. Okay, next one is capsules are most commonly composed of lipopolysaccharides, polysaccharides, lipoprotein, or polypeptide. And the right answer is option C. That is 2 and 4. That is polysaccharide and polypeptides is the correct answer, which are made up of capsules are made up of these two. Okay, so next one is which of the bacterial component are antigenic? Fimbriae, flagella, capsule, or ribosomes? And the right answer is option A. That is 1, 2, and 3. Fimbriae, flagella, and capsule. Okay, next one is bacterial flagella can be demonstrated by negative staining, silver impregnation method, gram staining, or dark field microscopy. And the right answer is option C. That is 2 and 4. Silver impregnation method and dark field microscopy. Okay, so move on to the next one. That is motility of organism is demonstrated by hanging drop method, Coulomb reaction, growth in semi-solid media, negative staining method. And the right answer is option B. That is 1 and 3 is the right answer. Hanging drop method and growth in semi-solid media is used for motility of the organisms. Next one is, and here we can see the Coulomb reaction. So positive Coulomb reaction where capsule swelling when tested against antiserum containing anti-capsular antibody. So in normal condition capsule is the outer coat, but in the reaction of Coulomb reaction the swelling of the capsule we can see, okay, by the attachment of the anti-capsular antibodies. Okay, so move on to the next question, that is which of the following organism produce spores? Bacillus anthracis, Clostridium tetani, Bacillus subtilis, or Bacteroid fragilis. And the right answer is option A. That is first three options, Bacillus anthracis, Clostridium tetani, and Bacillus subtilis are the right answer. Okay, so next is the spores are visible in gram staining, negative staining, modified Gill-Nielsen method, which is the acid fast staining, and Albert staining. So the right one is the option B. It is 1 and 3. Gram staining and modified Gill-Nielsen method is used for spore, where spores are visible. So next one is bacterial endospore are characterized by a low level of metabolic activity produced when nutrient supply becomes limiting, less amount of water activity, accumulation of calcium and diaminopicolinic acid. And the right answer is option E. That is all the options are correct according to regarding endospore, bacterial endospore. So next one is marked resistance of spore has been attributed to low metabolic activity, low content of water, impermeability of their cortex and outer coat, absence of calcium, and diaminopicolinic acid. So the right answer is option A. First three options are correct. Okay, so the last question is spores of organisms employed for sterilization control include Bacillus stereothermophilus, Bacillus subtilis, Clostridium tetani, and Clostridium welchi. And the right answer is option B, that is 1 and 3 is used as the sterilization control. Bacillus stereothermophilus and Clostridium tetani. Okay, so these are the all 25 questions of this series of video. Okay, thank you for watching this video.